All right, our next speaker is Malina Loeher.
Great. So thanks for the wonderful introduction. I'm a grad student at the Virginia Institute of Marine Science, and I'm really excited to share with you the first chapter of my dissertation. So I work on virulence, and as many other talks have alluded to, virulence is a really critical consequence of viral infection, and understanding virulence is really critical to managing diseases, particularly after host jumps. This is important not only for wildlife, but as we've seen over the last 2 years, also for human populations and many others. So we have a number of, we really want to know how virulence is going to change after it emerges in a new species, and I won't really go into this figure because I think probably a lot of people in this audience have seen it before, but in the very classic case study of myxoma virus in rabbits in Australia that was introduced as biocontrol, we saw that virulence attenuated after it emerged in a new host. And this field study really demonstrates what we call virulence trade-off theory today, where virulence as well as transmission rate and duration balance each other for a virus's optimal fitness over time. So if you're a little fuzzy on trade-off theory, that's okay, I have a slide for you. The foundation of trade-off theory really rests on the idea that virulence confers both costs and benefits to the overall fitness or R0 value of a virus. And a possible cost of virulence is that as virulence increases, hosts are killed or hosts die, and transmission duration is really cut off by this mortality. A benefit could be that virulence confers increased transmission rate over time. And with myxoma virus, it was more of a cost, and fitness, optimal fitness meant that virulence seemed to attenuate over time. So if we can understand the drivers of virulence evolution in this system and beyond, we can better manage diseases and possibly select for decreased virulence and disease severity in other systems. So I'm really interested in how these trade-offs occur and what kind of balances we can find. And I'm interested in exploring these questions in a different system besides myxoma virus. So the system that I worked with is infectious hematopoietic necrosis virus, or IHNV. It's a lethal virus in the Rhabdoviridae family, and it affects all members of the family Salmonidae, which is all salmon and trout and a few of their other cousins like char. The virus targets the kidneys and liver, and in aquaculture, it's really primarily transmitted horizontally. We care about this disease because it is a major pathogen for salmonid farming worldwide, and it's also a really big problem for conservation. So I mentioned that it has global spread now. It didn't always have global spread. It is endemic to the same approximate region that sockeye salmon are in the Pacific Northwest. We understand IHNV to have co-evolved with sockeye salmon, but as commercial industrial aquaculture expanded in the region, IHNV made a host jump from sockeye salmon into rainbow trout, and from then, from trout farming expanded pretty much wherever trout farming has gone, which today is across most of the northern hemisphere. So this is a globally spread disease with global consequences. I realize not everyone might be familiar with trout farming, so the background of this image is what a typical trout farm might look like. This image is from Idaho, where over 75% of domestic rainbow trout are produced in the continent in a pretty small region, which is really incredible when you think about it. And in general, can you see? Trout are raised in raceways, which are these concrete outdoor troughs that you can see at the bottom of here. And do I have a little? Yeah, so these are raceways, and as you can see, they're pretty large. Just for scale, this little red thing is a car. And even though this farm is in the same endemic environment as IHNV and rainbow trout, the actual environmental little habitats are very different than what a salmon or trout might encounter if they're in their wild habitat. So we think that aquaculture might have really different selective pressures, not only environmental, but also host specific. So when we think back to myxoma virus, this is a very different system. We're not only talking about terrestrial versus aquatic, we're talking about different kinds of hosts, mammalian versus fish. Myxoma virus is also vector-borne, IHNV is not. We could go on for a long time comparing different viruses, but the point is it's not really reasonable to expect that the same evolutionary pathways are going to emerge in every different system. Specifically for aquaculture, we know that high density of fish, low genetic diversity, growth rates, overlapping age cohorts, and pharmaceutical practices like vaccination can all have really big impacts on our hosts. And so we expect that IHNV might follow a different evolutionary track than we see in the wild. And we want to know what's next. And we think that we could be seeing diminishing costs of virulence in aquaculture, such that virulence might be actually increasing over time. And this brings me to my sort of main research questions. The first is that novel strains might be evolving towards increased virulence, that is after its host jump in aquaculture, and also that ancestral strains of IHNV are not following the same evolutionary track. So we actually know quite a lot about IHNV already. This is a cladogram depicting the genetic spread of IHNV in its North American range. And so this bottom half is what we term the U group, which is the ancestral group. And you can roughly equivalent this to endemic strains that are mostly correlated or mostly associated with sockeye salmon. And the top half of this cladogram is termed the M genogroup, which originated completely in aquaculture and represents a massive genetic diversification of IHNV after its host jump. Something this cladogram doesn't really show very well is the relative genetic diversity. We estimate that there's over 3 times as much genetic diversity in the M genogroup as the U genogroup. And the really exciting thing about this cladogram is that we actually have a lot of this cladogram represented in an archive of isolates. Because it's so important to conservation and industrial aquaculture, a lot of isolates have been collected over time, and we have tens of thousands of them in an archive maintained by the USGS. So for this project, we selected isolates that spanned both the top to bottom, the genetic tree space range of IHNV, as well as left to right, the temporal range of both the U group and the M group, and compared these in salmon and trout. Which is so cool to think about. Like, what if you had COVID strains from 50 years in the future? Here's what we did. So we conducted a virulence challenge. Each of these black boxes represents a treatment where the tank is the unit of is the experimental unit, and so each tank had a group of 15 to 20 fish representing a little fish population. Each treatment was issued either a low dose or a high dose of IHNV of one of 15 different isolates. And you don't need to read all of the names. You just need to know that some of the isolates were in the U group, that ancestral group, some of them were in the M group, and they spanned the temporal range of 1974 to 2017, which is indicated by the last little numbers of each of the isolate names. And I'm gonna keep color typing them by blue and yellow, so you can just kind of remember blue is old and yellow is new. So once we exposed our fish to virus, we maintained them for at least 4 weeks or until we saw a mortality plateau. We typically see that mortality plateau in the first couple weeks. This is what it looked like. We had a bunch of tanks of fish, very colorful, organized. And these are the results from the ancestral host. So I'm just gonna show you cumulative percent mortality today. So how many fish died over the course of the experiment. That's on the y-axis, and on the x-axis, we have all the different isolates that we tested. They are ordered first by whether or not they fell into the U group or the M group, and then within these large genogroups, they're ordered by the year of isolation, so old to new within their genogroup. And immediately you could see that we had a lot of mortality in all of our ancestral viruses, which is more or less what we expected because we understand them to be co-evolved with sockeye salmon. What's kind of interesting is that we don't really see much mortality in the M viruses. So viruses that went through a host jump seemed to maybe lose some of their virulence in the ancestral host. So this is great. This is our baseline for comparing things after the host jump. And to get our after host jump data, we repeated the entire experiment in rainbow trout. Same design, same setup, same isolates, same size. And the rainbow trout data looked like this. So same type of graph with cumulative percent mortality. And the U viruses have relatively low virulence overall, whereas the M viruses have pretty high virulence across the board. It's a little bit of up and down. But what I think is really cool is that, remember, we have isolates ordered from sort of old to new, left to right. And we see a pretty significant difference from the early ones with low virulence and the newest ones with really high virulence. And this trend was the same across both doses. So that's pretty interesting to think that there might be increasing virulence. And overall, increasing virulence, there we go. And so these are the same graphs that I showed before, just kind of smushed together with just the high dose data. We see that not only do we have really different trends in virulence between the ancestral and novel hosts, but they're also flip-flopped. So viruses that had viruses that were high in one were low in the other, and they're different from each other. So, as I said, virulence seems to be increasing among the viral isolates that are specifically associated with trout aquaculture, which is really concerning for that industry. And we also see that IHNV is exhibiting host specialization. So how does this fit into trade-off theory? So this is the same graph as we've seen earlier, but it seems like virulence confers more of a benefit in this environment and host system, and we're actually seeing virulence shifted to the right for increased virulence. And a natural next question might be, well, like, what about transmission rate? What do you know about that? How does that fit into this? Could that be the driving mechanism? And I would say that's a great question. So I also did shedding kinetic studies alongside all of these virulence assays, and I don't have the data for that to share with you today, but don't worry, we're working on it. And we're also doing a lot of transmission studies to study transmission efficiency of the different isolates and how those might have changed pre and post host jump. This is also just one piece of a larger team working on both full genome studies of IHNV and also epidemiological modeling. So hopefully I'll be able to share a little bit more about shedding and transmission at a later date. So this is my last slide. This is a non-exhaustive list of acknowledgements for my lab team, my committee, my close collaborators, Gail Kurath and David Kennedy, as well as my advisor, Andrew Wargo, and my funding sources. So I hope I can take some questions.
Thank you for that wonderful talk. We definitely have time for a few questions.
Hi, great talk, and this is a really cool system. I was wondering if the virulence of the different strains on their actual hosts, so the virulence of the ancestral one on the salmon versus the virulence of the new one on the trout, are those virulences actually different? Or are they the same? And so actually this increase over time in the trout is more due to just adaptation to littoral trout versus adaptation to the different agricultural ecology of trout farming. Does that make sense?
I think that makes sense. So you're asking kind of what can be attributed to host factors versus environmental factors? That's a great question.
Or just does this pathogen in general just like to be high virulence?
Yeah, I think you'd have to know a little bit more about what's happening in the wild, which is really difficult to study. But in addition to doing dose and host treatments, we also did a temperature treatment for this experiment that kind of bridges the gap between wild salmon environmental factors and farmed trout factors. And we didn't really see any differences from that treatment. So we concluded that this is really more of a host specialization issue. It is possible that it just likes being a high virulence virus, but that's kind of difficult to say without knowing a lot about wild salmon and trout in the 1950s and 60s. Okay. Yeah. Does that mostly answer your question?
Yeah.
Okay.
So with low mortality, does the ancestral strain then protect the trout from infection with the trout strain?
That's a really interesting question. I don't know, but I would guess maybe. I think if we could do some co-infection studies or sequential infections, that would be really interesting. But we also have thousands of isolates to choose from, so you'd have to think a little bit more about how, which isolates would you choose and why? Because a lot of these co-circulate, a lot of them are found in overlapping regions and even in the same raceways of trout. So the short answer is I don't know, but that would be really cool.
Do we have anyone's online? Nope. All right.
Hi.
Maybe just one more, yeah.
Sure. Fantastic talk, amazing system. One thing I noticed on your cladogram is that the U genotype is actually, it looks like paraphyletic. So did you include that subclade of that is actually in the, that should be in the M clade, I guess? Did you test those? And
so yeah, The phylogeny of the system is really cool and really complex, and I actually don't understand it super well. So I can point you to some better literature that would really like get into your question. The short answer is no, we only considered one major group of the euclade called the UP clade or UP genogroup because we think that all of the M's came from that group. So there's a lot more genogroup complexity and substructure in the M group, and so we just included the ancestral group that we thought was the best representation of where the host jump probably occurred.
Yeah, cool. Thank you.
Thank you, Malina.